Hi, welcome to Health Horizons, the podcast at the intersection of health, tech, and policy. My guest today works right at the frontier of AI and medicine. She built an AI tool that reads an ordinary mammogram and flags a woman's breast cancer risk years before it appears. She's used machine learning to discover brand new antibiotic, and she's now building systems that predict which cancer's treatments will work for which patient. Regina Barzilay is professor at MIT, the AI faculty lead at the Jameel Clinic, a MacArthur Genius Grant winner, and one of Time's 100 most influential people in AI. But really, her work didn't just begin as research. It began with her own diagnosis. In 2014, Regina was diagnosed with breast cancer, and after treatment, she turned her focus toward the clinical AI that she's known for today. So when she talks about patients, she's really speaking from the other side of the scan. Now, this episode was also a personal one for me too, and that's because Regina is one of my research mentors. And a lot of the questions this show is built on are ones that I first started asking because of her. So I hope you enjoy. Hi, Regina, and welcome to Health Horizons.
Great to see you, Mel, again, even remotely, and happy to be here.
Thank you. A consistent focus throughout your work has been personalization of care. So you've developed cancer risk prediction models, which enable personalized cancer screening strategies for patients. And some of your current work today is more focused on personalizing treatment for cancer patients. And so what I want to start with is assume I'm a patient in 2036 with an undiagnosed early stage cancer, and personalized medicine is quite broadly available. I want to understand how my patient journey differs from the patient journey that I would have today.
So first of all, I hope that in 10 years, you're not going to have cancer. Nobody's going to have cancer. My view is that the first way to change this journey is actually to diagnose it at much earlier stage, that we are not going to be seeing as many late stage patients as we see today. And a majority of people can, first of all, identify it in a very early stage or even have preventative treatment that would not put them in the cancer journey in the first place. But if one already is on the journey, what I hope we will have are treatments which where we are not guessing where they're going to be working on the patients, where we have much higher certainty what will be the outcome of the treatment, and we will have treatment which would have much less side effects, because what happens today, one may treat cancer, but they may obtain many other diseases as a result of very toxic treatment. So again, just to summarize, really early diagnosis. Second, eliminate uncertainty regarding the efficacy of the drug, which really is tailored to your profile. And third, less toxic therapies.
And when you talk about these, what kind of data do you need to make that happen?
So one interesting point is a big difference between two ways to look at the patient. If you are in normal healthcare system, wherever you are located, there are standard sources of information that you would collect about the patient. You would do some radiology, some type of imaging. There will be some pathology slides typically associated with extracted cancer. And there will be some molecular profiling. Like, for instance, in breast cancer, one would predict whether they have HER2 or whether it's hormonal positive or hormonal negative, but very limited characterization. On the other hand, if you're looking at research papers that publish about cancer, there the level of information that you have about the patients are very, very different, and they are not tractable today, and I'm not sure if in 10 years it's going to be different, this type of information to be collected in clinical care. For instance, single cell sequencing or spatial transcriptomics. It's slow, it's expensive. Again, who knows, maybe in 10 years it's going to be very cheap, but still it's very, very detailed biological data about the patient. And the happy part that what happened actually in the last maybe a year or two, there is a realization that you don't absolutely have to have this very detailed and expensive molecular information. If you have it, it's great. But if you don't, which happens to normal patients, you can glean a lot of kind of molecular profiling of the tumor directly from the information that we do collect, be it pathology slides or be it blood and other sources. So what I can imagine, again, if we're looking in the frontier from 10 years, I would acknowledge there will be more types of assays that are available, more blood diagnostics and other. But even if we're staying roughly within the same realm of information, we will have methods that can extract a lot of molecular patterns from media that are not traditionally thought as molecular media, like pathology. Let me give you an example of what I mean, because one way to think about it, there will be more data. Maybe spatial transcriptomics in 10 years would be a trivial thing to do because technology is going to be better, maybe. But even without going there, today, when we're looking at the pathology slides, what pathologist does, they would characterize cancer, the stage, the grade, some molecular features of the cancer. On the same time, we developed at MIT recently a model that can look at the pathology slide and extract for every patch on the pathology slide what gene expression programs are active on that slide. No pathologist would even try to do that because they can't. Maybe for some of these programs, like for instance, there is one that corresponds to blood vessel expansion. Maybe this one you can sort of see, but there are many others that you can't see, with your naked eye, even if you are a specialist. But machine can. A machine can identify all these different pieces. Why they are important? Because they give you a very detailed description of what's going on in the tumor. And depending of what kind of processes are already in place that drive the tumor, you can already, in a more direct way, select which treatment may come next. Or maybe you can see that whatever you are planning to put the patient on is very unlikely to work because of this detailed molecular profiling that happens. And connecting it to another piece that we didn't talk about, which is drug discovery. When we're talking about personalization, the discovery is really important. It is not enough when you have the pathology and you are characterizing something, you still don't really understand what will be the target for the next treatment. And through this type of molecular characterization, you can actually identify and say, all these non-responders are characterized by this type of molecular programs. Maybe we need to develop drug which operate exactly on this program. So you bring the patient journey together, not only in terms of diagnostic, but also identifying something that will tell you what will be the next generation of drugs. But as I characterized to you, that even today, this model can identify something that human eye really cannot discover. The question is what the doctor will do. And we know that at least today, the clinician needs to operate within the standards of care, within particular protocols. It's not like even the machine tells, oh, I see these programs, what do you do? And there are different approaches that we can imagine. We can imagine that there is 10 drugs or 20 drugs from which you can select, and the machine can just give the likelihood that the patient is going to respond. So the doctor can make more informed decision. Maybe if the machines are going to be much better, maybe you can even imagine giving a drug which was not directly developed for this particular type of cancer. But you can say, can we trust the machine in doing even the first selection among the 10 drugs? And I had a lot of conversation with oncologists, who are my collaborators, and they're saying that, especially when we're talking about metastatic cancer, for many conditions, there are already multiple drugs on the market. And to a degree, it's a matter of guessing. I mean, there is a general, let's say you have, EGFR, but the different mutations, the different drugs may work better. But there is some element of guessing when you're giving a particular drug. So the way I can imagine machine learning and AI models coming in, they can decrease the amount of guessing due to this inherent uncertainty. And of course, it needs to be rigorously evaluated to ensure that we're making better decisions.
Lots of topics there, and I want to deep dive into a few of them. And so I think it's worth diving into kind of the landscape today then to give us a better sense of what's actually being done in terms of these personalized treatments. And once we have that sense of the landscape, then we can deep dive into what you started talking about, which is your current research on these personalized treatments. So to start off, what is the state of the art around these personalized treatments today? And what are their limitations?
So if we are thinking about, how medicine is run today in the past, that most of the time to show the efficacy of the treatment, you need to do randomized controlled trials where you are giving a patient with similar condition, one arm gets treatment A, another treatment B, and then you come to the conclusion which one will be more effective for each group. So before you start doing the trial, you need to define in a very clear, deterministic way, what does corresponds to this cohort. So you are already, to a degree, removing any ability to look at the probabilistic set, and you need to decide about the cohort and characteristic of the cohort. Then after you've done that, you're kind of going to look at the treatments and identify. And if you look at any trial, even like great efficacy trial, it's not like the new treatment works to 100% of the patient. And sometimes if you look at New England Journal of Medicine, one trial that I remember I was looking at in detail for personal reasons was a trial for using a various suppressant for premenopausal breast cancer patients. If you look at the efficacy between the arm that received this treatment and the arm that didn't, the difference is tiny. And the question is, would you give such a serious treatment because it has multiple side effects? Because somebody clearly benefited. We just don't know who. And the choice is that, the clinical association is to decide, how much should be the difference. And even if the difference is, like, say, 10% or 15%, still not everybody. But we cannot really today say who are the one from this cohort who are going to benefit and the ones who are not going to benefit. We're going to say they correspond to cohort. Overall, this group of patients did better when they got treatment A. We're going to give them all treatment A. So you can see that there are lots of patients in this cohort that are unlikely to benefit. Maybe they don't need this treatment, or maybe they need another treatment. But the way the trials are run, you are absorbing this kind of sacrifice in order to make empirical comparison between arms. So that's what happens. That again, if you look at, I'm familiar with a number of areas within, but let's say breast cancer, there are several cohorts. There is hormonal positive, hormonal negative, HER2 positive, HER2 negative. So there are several cohorts, and each cohort, roughly speaking, is recommended the same type of treatments that once in a while is updated if a new drug on the market. What I'm describing to you primarily goes up to pre-metastatic disease, because there is at least some homogeneous way to characterize it. When you're moving to metastatic world, it becomes much more complicated because the cancers are continue to evolve and they look quite different. So there is some element of guess which one you're going to give. It goes even beyond this cohort that I described, because there is so much heterogeneity in the disease that at the end, the clinician needs to make a guess what may work on the patient. So this is pretty much how the field operates today.
And so what is the research that you're doing then to try to address this uncertainty?
So the research that, I always wanted, I felt very unsatisfied that my group works primarily like on risk assessment. It's a very important topic, but there are a lot of people who are already sick and you really want to help them, but you cannot help them because they follow whatever is prescribed to them. So I was very lucky because we are part of ARPA-H program, which is called ADAPT, where the person who started this program, Andrea Bild, she's a faculty at City of Hope. It's a hospital in the United States. So her thought was, let's look at metastatic patients that already fail the first line of treatment and identify what will be the best treatment we can provide for these patients among different options. It's not like anything is go, but the clinicians would say there are 10 possibilities. Let's identify which one is going to be the best for a given patient. And they currently run 3 clinical trials, one in lung, which happened to be in City of Hope. Actually, the second one is in colon in MD Anderson, and the third one is UNC and Yale, which is breast. So you have these cancers, which are really, the cancers that affect lots and lots of people. And again, we're looking at these patients who are way in their trajectory, of treatments of metastatic disease. And we're trying to recommend for each clinical trial, the clinician identify, different arms of treatments, and we're trying to identify it. And going back to your question, what information do we have? We have the imaging, like for instance, in lung, it may be a CT scan or PET scan. We have their pathology slides for the first diagnosis, because in the United States, you only get pathology after the first line of metastatic disease. But we have their pathology, and we have ctDNA from these patients. And also there is something which is not typically part of clinical care, but here they agreed to do, which is RNA-seq, which is a sequencing of the tumor. And this is the information that we have based on which we need to recommend the treatment. And the trial's already enrolling patients, so it's underway.
And so if I understand correctly, so you get all this data from the patients who are enrolled, and then your actual research recommendations then are applied to the patients directly.
Yeah. So of course, we train it using other sources of data, but once, everything is aligned, you can use all or some parts of the data to make recommendation. To select among the set provided by clinicians which one of the drug regimes you're going to give to the patient. And also in some cases, we want to predict as soon as possible that the patient doesn't respond to treatment, because it takes a long while today to see it. So as early as possible, you understand that they're not responding already, that you can switch the medicine.
And the project started, maybe it started 6 months ago now.
It started exactly a year ago. It was a very stressful year for us because we didn't work before with RNA-seq. We never worked with pathology slides. We did work with big imaging, but here we have brain imaging and all other, like there were a lot of ctDNA, all this, there were a lot of new modalities. And we really spent a lot of time hunting for the data to train our models. And this was a research on its own right. But now a year later, we actually have seen many of these models start working very nicely. So it was very exciting.
Could you talk me through some of the other challenges, like research challenges that you've been facing in the past year then?
Yeah, so let me just say one sentence about, besides the modalities, about the data. Like, we made a lot of progress. I'm talking not MIT, I took in the whole research and medical community, made a lot of progress on, primary cancers. And for instance, in breast, if the patient is diagnosed with a local disease, the likelihood of 5 years survival is like high 90s. Not the case if metastatic cancer. The outcomes of these patients are not very different than they were 5 years ago or 10 years ago. So you can say that these patients are the one where we should be really investing a lot of our resources to help them, because we still want, of course, to improve our primary care kind of cancers, but those are the patients where we really didn't make the progress. And the most bizarre thing that I discovered, that among many data sets that you can find about cancer, there are many data sets, very, very few data sets have data for metastatic patients. And what we see from various experiments, that their cancer is very different from the cancer on primary. You can say, maybe I will train on primary and bring it to metastatic. It doesn't work this way at all. And there is one data set from Hartwig, I should say, from Netherlands, which is maybe the only one which is dedicated to metastatic patients. There are some tiny ones, but really very few pieces that are outside and that are available for training the models. And this is very upsetting.
How do you explain that? The fact that there's so few data sets?
I think that, these data sets, they are currently not the fact that there was like a concerted effort of oncology community and say, what are the areas where we really need to have more data because we didn't make any progress using our traditional tools. Let's go and get this data. Let's pay somebody to make this data. It just happened to be so that, there is some conversations and, certain group of people will decide they want to put this data public. I will give an example in US. For instance, there is this data set, which was excellent, that we use for training SIBO, which is called NLST for lung, NIH, NCI, Paul Pinsky, particularly Dr. Paul Pinsky. They put effort, they created this invaluable resource that all the community is using for developing the models. On the other hand, there is no equivalent of that in the United States for breast, which should be much easier because every single hospital in the country is doing mammograms. So there's no good explanation for that. It's just somebody felt the need, or it's aligned with their vision, and they put this data set out in breast, which is very important. There is no such resource. And I hope that somebody who is listening to our conversation, there should be a change in thinking in the public system when we are investing in research, that we have much more targeted for the goal approach to which data do we release. And I would give you some examples actually, where there is a huge effort in producing some data. I don't see any value at all. And they continue to produce it. So the question is, again, instead of saying I have an instrumentation or I'm close to the hospital which can release it, let me just put it out. This is not the right approach. And unfortunately, many times that what happens, and we need to change it, because if we don't have data about metastatic patients, we're limited in how we can help these patients. So this is just a comment about what we can do. But to me, the questions, the research questions, were the following: that even forget for a second that we didn't have much data, you need to be able, to create your stone soup. That you don't have a perfect data, we may not have it for many years. How you can learn something from the imperfect data. And let me go to this data set that I was just talking to you about, predicting how the patient will respond to the treatment from RNA-seq, from the expression data. And you can see, for those of you who don't know, it is just an array of 20,000, values that record the expression of different genes. So you have cell lines, for instance, and you have patient data. Patient data is limited. Cell lines is typically very large, and you can, for cell lines, you have, cell line with a particular expression. You put different drugs, you see what happens, and then you see the response. So there are several changes. First of all, you need to learn it, you're learning it on cell lines, but eventually you want to bring it to humans, correct? Because cell lines do not represent, they're a proxy for human cancer, but they're not truly human cancer. Another problem is that even the basic question, how do you encode RNA-seq? How do you take 20,000 expression and encode it to the machine that is like biologically meaningful? How do you take, let's say, small molecule, which is a drug, how do you encode them to properly capture the interaction? And, when we entered this problem, said, oh, this is easy. And this is a work of my student, Serena Ku. We did an obvious thing. We say, oh, let's just do some embedding, which means that, you kind of do some dimensionality reduction on this RNA-seq, and let's just encode the molecule, like let's say you can use GNN or something, and you feed into neural network and you see what happens. And what we discover, nothing good happens. The numbers are really low for various reasons. First, one obvious reason is that, you don't have millions of drugs, correct? The number of cancer drugs is still very limited. You don't see enough interaction. This is a very sparse data. It's not like a normal data that we see in computer vision that you can just kind of do an obvious architecture and run it through. So this obvious model didn't work, and it was not only our discovery. There are people who published it. It's like, it's nowhere close to be useful for anything. So we said, okay, the data is small. You cannot directly extract the pattern, again, because the representation of molecule is small. We need to think how to take this data. But clearly, it's not like every molecule would work on every cell line. There are still patterns. And Serena had a very interesting idea. She thought we need to directly model biology, because the way that this RNA-seq that represent the state of the cell and the molecule interact, there are actually known biological mechanisms by which the things interact. They're called pathways that, when molecule interacts, it activates certain thing and it sets the chain of reactions. And in biology, people spend significant amount of time understanding from the biological viewpoint what are the different pathways that can be like a chain reaction for the interaction. So this piece is well understood. So the way she translates that if we know already something about this pathway from the core biology, maybe we shouldn't make the model because it doesn't have enough data to learn everything from scratch. Maybe we should learn how to map this problem, how to reformulate this problem. So now this interaction, there is like this vocabulary of all possible pathways. And we cannot make an assumption that you would know, every single one of them, but you can go from the RNA-seq, from the cell or from the patient, map it into the pathway language, and then learn the interaction between the molecules and the pathway, which make it much easier to learn. You are reducing from these 20,000 things that, we don't know which one are relevant, to a subset of this known interaction. And then you are learning the interaction between small molecule and these possible mechanisms that you are much more explicitly kind of exposing to the model, the mechanism, and then you can learn from smaller amount of data. And then another piece, how do you even encode small molecule? Again, we typically in our previous tools, when we did work on small molecule, we use graph neural network, other people use, smiles transformers, whatever. Now, from this representation to learn, again, you need more data. But Serena had another beautiful insight related to our work on bolts. She saw that what we actually care about is how this molecule interacts with various proteins. So maybe, and we know what are these proteins that, drive this behavior, not only for this drug, for all, there is a set of proteins that are active in this pathway. So maybe we can just run bolts and represent the molecule based on its interaction with the core proteins that are involved in the response or in cancer progression. So you again exposing the behavior, making it easier to learn. And from there, not only that she predicts the response way more accurately than anything else, but she identifies what are the pathways that are involved in response for a specific patient. Because for different patients, the same drug, depending on the patient, may do different things. And the next step is actually to take this tissue, go to the lab and check if for this cell line, this is what exactly happens. So you can really get to biological interpretability of this interaction. And this was, again, how do you rethink the task? Because you have limited data, which, puts a lot of constraints on you, but you need to rethink the model with the biological constraints. Again, not just to directly build on the biology, but learn the embedding of the pathway to translate it into a new language, again, utilizing the main specification. So this is just one example.
So really interesting. So this context of limited data has led you to rethink the approach.
They rethink the model. Yeah.
And I think what's quite interesting as well is sometimes there's this idea of a black box model. It seems in this case that the interpretability piece is also very important through the understanding of the pathways as well.
Yeah, and I'm very skeptical about interpretability in general because, like, if we do not understand it, like so many times, when you're predicting, like on Mirai and SIB, you're trying to predict, and even if you show what the model looks at, you still, as a human, cannot know. But in this case, they have actually a very clear way to verify the pathways that the model identify. You can just do biological experiment, not we, but our collaborators, they can do the biological experiment and then can tell, is the reasoning of the model biologically correct or not? So in this case, interpretability does make sense.
That connects to something I wanted to ask you about because obviously this type of project is very unique in the sense that usually when you develop a tool, it takes years for it to be applied to patients. In your case, it's directly applied. It'll be applied in the next few months, essentially. So extremely quick. And so I wanted to ask you about that validation question. When a model is just used for patients in such a reduced timeline, how do you think about validation and making sure that the models work well?
Yeah, this is a very interesting question because it is true. And this is one of the remarkable things about this program. Again, its name is ADAPT. And for those of you who are listening us, you can just go to ARPA-H ADAPT and they have like a beautiful web page that kind of describes the rationale behind it. They work together with FDA to enable these trials because we need to ensure that they're safe. And this was one of the requirements of the program, that if you are claiming that something happened, you need to have the biological justification for why is it happened. So whatever you are predicting, one way you can do it, you can set up some population and run on it. But another way, which is like that you need to convince yourself that it was not just fluke that things look good, you actually go and you validate it in the lab. So I think it's really important. It gives confidence not only to us, but to clinicians who actually are prescribing these very heavy treatments to patients, that there is a rationale behind these decisions. So this was actually one of the things that the program required, is to provide biological interpretability to the decisions.
Is it something that keeps you up at night though these days? Is that something you're slightly worried about?
I am really worried when I'm thinking, and like the statistics that I like to say that, depending where you go, but it can be as high as one third of death in the United States, at least, is because of medical error. So if you talk to any oncologist, they are totally aware that when we're talking about this advanced disease, there is so much guessing involved. And what I'm always thinking that if you have like a primary, let's say breast cancer, it doesn't matter where you go to the top research place or you go to a rural clinic, there is very clear guidelines and most likely you're going to get the same treatment. Most people will get the same treatment. But when we're looking at the metastatic cancer, we can really see there is a huge variance on the survival. First of all, it's a cancer, but another thing is what kind of treatments actually patients get and how well the clinician was able to find one which works for these specific patients. So we're not comparing to the ideal world where everything works. My understanding, and that's what I understood from not only clinician who works with us on the trial, but many others, pretty much every clinician has in their head some list of options and they have some heuristic to select. So my question is not whether we're going to cure the patient. My question is, can we do better than what they're doing now? And I think that adding rigorous way to select is a step towards improvement. Do I feel that we really need to work extremely hard? And Mel, how much hard work happening in the group, because I want to
Yes.
make sure that we are really doing that most effort in making it as perfect as possible. That on our end we did everything we could. I'm like right now I sleep well, but until very recently I wasn't, because I was really stressed if we can really deliver. And the data, and if I may say something, which is about what regarding not keeping me at night and related exactly to this work of Serena, one of the things that when we started working, the RNA-seq, again, it's an array of 20,000 values. And the question was, how do you encode it? And even for those who are not computer scientists among us, the word foundation model. And if you're working like in language, you would always use foundation model to represent, the sentences or the words. If you work on proteins, you would also use foundation model. So, and it became such a common word that you say, wow, so the thing that I should be doing is taking my RNA-seq with this big array and representing it with foundation model. Again, what is a foundation model? Is a way to do dimensionality reduction in such a way, not just randomly to kind of reduce the dimensionality, but to map it into some new vector space so that the distance between the representation in the space really corresponds to similar patients, because it could be the two states of the cells look different based on 20,000 numbers, but in reality they correspond to the same thing. And there is like a principal mechanism to reduce them into some representation which captures their biological similarity. And that's so, like in proteins or like in language or in vision, they're foundation models for RNA-seq. So when we started working, we said, what can we do? The question is, of course, let's just take one of this foundation model, and God bless, there are plenty of them, and and encode it. It was like a standard step. And when we've done that, it didn't look good at all. And we were very confused because there is like continuous effort in Arc Institute at Stanford and other places which are working on creating more in single cell RNA. There are lots and lots of this data out there, and people continuously publishing foundation models. So I was thinking, okay, maybe this one doesn't work. Let's try another one. And it didn't work. And again, because you're developing a very big system, you say, maybe it didn't work because our data is wrong. Maybe there is something wrong in the pipeline. So because one of the challenges when you're debugging the system, there's so many different places it could be wrong that you are thinking always there is something there. We didn't even question ourselves, and the results were not converging, and it was going on for months and months. And I couldn't say that I really didn't sleep at night, but it was really troublesome for us because we just couldn't understand why it's happening. Then we saw a paper that was published that kind of said that, for some task, I don't remember what was the task, but if you're just using directly the 20,000, it works better than the foundation models. To me, it was like a bit of a red flag. And then afterwards, we said, okay, let's just go and use a representation which people use before foundation model, before deep learning came in, which are very simple approaches. Like one simple approach, you just look again, you have these 20,000 genes and you have your population. Say, let me just select 200, which vary across the population the most. It's called highly variable genes because many values are going to be exactly the same. So it's like an heuristic. You say, let me just look at those. Or there are some sets which are called gene sets, which people through biological knowledge identify this is an important subset. So the heuristics that people use pre-deep planning. And to our surprise, this heuristic work way better than the models developed for this stuff, for foundation model, like but by not by like 0.5, but like consistently across many data sets. And then I thought, okay, let me just look at the papers because these papers continue to be published. Let me just look at the models that are developed specifically for response on drugs because maybe there is foundational model for other purposes. There is, for instance, a paper from Harvard called Compass, which developed this predict whether the patient is gonna respond to immunotherapy. Again, we're taking this paper and we take their representation on their data set, not even now, that's it, on their data set, and guess highly variable genes are much better. And that's what I was saying. And another interesting aspect is that if you look at all these papers that do this foundation model, none of them actually compared against this simple heuristic. And that was upsetting for me for the following reason, because we really spent several months trying to debug and understand what was wrong until one of the students had the idea, let me just use something simpler, and we saw the difference. And again, that there are more data is collected, perturbation data. And the question that I'm always asking, can you show me like a biological discoveries that you got from this foundation model? Can you show me a case where, you know, like, let's talk about bolts for a second. There are lots of people who are using bolts. We don't need to advertise bolts. People just plug it in and it works, correct? So, and there are lots of discoveries of people who are not computer scientists, who are biologists, who use this piece, and it helps in Ladybus AlphaFold and many other tools. So here, can somebody point me to a piece where it's actually for real a discovery? And here we've seen, and now we've actually completed publication about it. And the reason I never wrote about negative results papers, but I think the goal here is just to tell to the people like me, don't waste your time. And it's a very interesting question that I have some hints as to why it didn't work. But this is an example that we invested a lot of this data. One can say, but if we maybe continue, doing more, maybe it will come up. But I think at the end, the money for data and for model development is limited in this world. So before we are investing in a certain data collection effort, or model development effort, we need to have some confidence that it's going to be useful to the patient, because at the end, we are not developing an algorithm for the sake of algorithm development. For this, you don't need data. You can just create a beautiful mass. This algorithm we are developing to actually help in therapeutic applications. And if we don't see any value, maybe we should be looking at a different type of data.
Right. Yeah. So real question, not only about just collecting data, but just making sure that you collect the right type of data.
That you collect the right type of data, and another conclusion is, especially in this field where there are a lot of people who don't have training in AI, the kind of computer science training, that we are very rigorous, that whenever we put pieces together, that we understand exactly how every piece computes, and not to put the piece because it's looks pretty, it has the word foundation model, but because it really improves. And again, we've seen in many areas it does, but we need to be totally honest with ourselves, because across all of these models, they're not 99, they're very far from 99. So unless we very honestly as a community say that's where we are, this is maybe the area where we need to focus more to make it better. But unless we understand where things are not working, we're just going to be continuing to build on flawed foundations.
I want to slightly shift gears and talk about how what you're doing in the context of this project could be extended to more healthy or patients at the earlier stage. So, as you said, currently it's applied to patients who very unfortunately and sadly have really no other option. And as you said, it's a lot of guesswork. But I imagine applying this methodology to patients who are just at an earlier stage could also reduce some amount of guesswork for them as well. Do you think we should move towards just generalized, personalized treatment recommendations then?
Yeah, absolutely. So first of all, like I kind of glance and talk about the pathology work, and this is a work of student Aziz and my student Gabriele. They actually look again by looking at the pathology slide, they kind of understand in a very detailed molecular way what goes cancer wise. And this is really important even prior to the fact that the cancer become metastatic. Like when the patient get a primary diagnosis, the biggest question that pathologist needs to decide, and that what on the back of the mind, both of the clinician and the patient, is this patient likely to metastasize, meaning that they are not going to respond to the treatment and they're going to get this non-local disease. But the question is, can you using the pathology slide, because you understand what happened there molecularly, to predict how the patient will progress? And this is really critical in treatment, because if you can identify this small cohort of patients which are, have high risk of developing disease, you can be much, much more aggressive in the intervention. At the same time, if you can identify the patients that are unlikely to go this route, you can totally change their treatment. And I would give you an example of a personal story relates to my own treatment. That after I finished my cancer treatment, I was placed on a drug, tamoxifen. Just after I got through the treatment, the recommendation changed. Previously, you have to be on tamoxifen for 5 years, and the recommendation was now to be on 10 years. Again, there was a clinical trial, and the clinical trial demonstrated that, it does increase survival. This is not a question. But again, we didn't really know for whom it increases survival. So we gave it to all the patients that were in this group. And I clearly remember, I mean, I personally didn't have many side effects of this drug, but I clearly remember that when I was, on 5 year mark and then later 6 year mark, I saw some paper came out and said if you take it 7 years, it's exactly like 10 years. And it was all very much not personalized. I just was afraid to get off, and they took it for the whole 10 years, like to the day. To the day. And you can imagine that not everybody needed 10 years, but because we didn't have any other option, I had to do it. And many other patients are in the same boat. So if you could look at my sequence of my tumor, or you can look at my slide and understand exactly what will be my trajectory, then I didn't have to be 10 years on the drug. So these type of questions, I think personalization is really important. Let me give you another example. I don't have a solution for it, but we're actually looking at it with Bolt, which is very interesting question. When you as a patient get, and it's not only true for cancer, it's true across medications. When you are getting to a serious medication, typically the doctor would give you a long list of possible side effects and say, we don't know what you're going to get. Like, I still remember, I got like pages of different side effects. At the end, I got a side effect which was not on the list. And there is no understanding how, you as a patient are going to respond to the drug, because sometimes the side effects can be really severe, and it is a factor in deciding which medication to place the patient on. And let's say if we know that this patient would not have side effect in the drug, you can increase the dosage, would increase the efficacy. Or if the side effect is to come, if there is really a chance of getting a side effect, then you can mitigate it, that you don't need to wait to see it erupting. So again, we don't have capacity truly to predict who is going to get it. So personalization, even in this case, can totally change the game. I hope that we would be able to help with it. But again, going back to data, today I didn't find any data set which really clearly has, the treatment, the biological data of the patient and the side effect that they encountered. And many times the data is not even properly collected, because unless it's severe side effect, it's not even reported to the doctor. But you can see how data determines what kind of questions we can ask.
And when we talk about the personalization of treatment, so it's understanding which drug will work best for which group, understanding the side effects, as you said, but there's also I guess a part of it is understanding which existing drugs are going to work and whether any like new drugs or combinations would work. And so we enter a territory of lots of uncertainty as well. And it raises lots of questions around regulation when you have much more personalized drugs for individuals or smaller groups. How do you think we should shift from this one size fits all approval of a drug or a device towards a much more specialized approval? What needs to change on the regulation side?
So right now there is a very clear, deterministic description, correct, of what is the scope of the patients that the drug can be given to. We know that sometimes the drugs are used off label. But as we, are getting to this personalized world when there is a test and the test predicts something, so there are already changes in the regulation. There are some tests that are approved, like for instance, Arterio's test for prostate cancer, drugs for J&J drug. They take the pathology slide, they predict the likelihood of response. So even though the drug was not improved this way, but in the clinical guidelines, this test that can be used to decide whether the patient needs this drug. So I think there will be a parallel line, which currently is not very strong line in the regulation, but really the development of this new diagnostic, which will be AI-based diagnostic, that to a degree is going to be tested separately, but it will be some black box to a degree, which is separately approved and tested, which, I don't know, takes, let's say, pathology of the patient and makes a recommendation. So there will be a way, hopefully, to run this test as a device. So it needs to go through that. And then it will be paired with a therapeutic efficacy. So it brings us to the question that we need to better regulate devices, in this case, diagnostic tests, and then pair them accurately with a therapeutic. The real question is that we know that AI develops much faster than the therapeutics. Today, still, despite all the advancement due to clinical trial, it's years in development, while the new diagnostics comes about very fast. So the question is, how do you really, work with these different speeds of development?
And if there was one policy change or one policy ask that you think could have a great impact on the timelines or on how this is done, what would it be in terms of policy?
I think right now, and I don't really know how to formulate this policy, but right now there is nothing that requires a clinician. The drug is approved for certain population. There is nothing that requires you to use new diagnostic or new personalization tool, even when these tools are developed. You don't have to do so. You can just continue whatever guidelines you are using. So I think that really streamlining and making it easier for the doctors to adapt, and then not only to make it easier but also to adapt this new personalization models. This is something that should really be brought into the attention, because the problem is that when we are not using the good personalization tools, not only that it decrease obviously has negative effect on the patient, it of course increases the cost, because now you put them on the medication, it failed, and you put them on another medication. So how do you really create a safe space for translation of this technology and require them to use it? This is something that we need to think from the policy perspective.
I want to take a step back from the technology and focus on your journey. So before developing AI tools for the clinic, you'd spent years using AI to decipher lost languages, and then you got diagnosed with breast cancer. So could you take us back to that time and share how you experienced that?
So I was using developing models for a variety of things besides ancient languages. This is maybe the one that is still remembered. But it's an interesting point that when many years ago, in like 94, 95, I started doing NLP. It was an interesting time, because the field, like today, it sounds like unbelievable tale, but at the time, people still believed that you can do, language processing by writing rules and grammars and whatever. So when I entered the field, it was just beginning of the change. I mean, the change was already happening, but there were still kind of coexisted both community. And I remember when I went to the first conference in 97, I saw there were half, I don't remember the ratio of people who were still doing these rules things. And there were already the first statistical and machine learning models that were, changing the field. And but by today's standards, these models would be like really baby. But I was really shocked, saying, wow, you can do it without writing rules. You can do statistical things. We didn't even use the word machine learning, is like statistical model, how exciting it was. And what was interesting that within really few years, Google created, machine translation system that, originally it was a bit brittle, but the field totally changed. I am not even talking about the current, language models, but field really changed. Like there was this huge change. So when I came after I was diagnosed with breast cancer and I started my treatment at MGH, this was really bizarre that every single point of treatment, from diagnosis to deciding which procedures to take, everything took, it was really old times. There was really no progress. And I talked to some women who were diagnosed a decade before me. They went through exactly the same pipeline. And I was really confused because my office at MIT is located across Cork, which is a cancer institute. So, these people are working every day on something. MGH is located just, one subway stop away. Where is it going? Like all these drugs were there forever, the same diagnostic pipeline. What's happening? When there was not AI, there was no data science, there was no data. And people always were they writing it by hand? No, they were not writing, they were typing in the computer. There was electronic medical record, but this was the end of technological development in that area. So when I came back, I decided that I want to do it. I mean, I had like a really strong, I didn't want to do any NLP. I saw it's like a wasted time. And the reason I saw this, so there are many people who are doing it. It doesn't matter if it's me or it's somebody else. It's like a big developed community. And I was even angry at myself that I was working on ancient languages because I saw people who are really suffering. And the world of MIT and the world of MGH, which is like 20 minutes walk, are really unbridgeable. You see true suffering, which we don't see at MIT. We see that somebody suffers if they didn't sleep at night and they were doing their homework, or if they didn't get A. It's a suffering. And I don't want to be disrespectful or feeling. That's how they feel. But there you see really people who are fighting with everything they have for their life, and you see exhaustion, you see lack of hope, you see the fame. It's really tragic. And I wasn't, when I'm thinking, I was like 43, so I wasn't that young, but I all of a sudden saw a different world that I was not exposed to. And I thought I want to do something. And it was not an easy journey because at the time I was not super popular and people were not standing and, Billy doctors and saying, oh, this is, Regina from MIT. That come to solve our problems. Many of them thought that they're already doing great and they have real things to worry about, which is taking care of their patients, and you come and waste their time. And some of the thoughts that originally I have were really not good problems. Like, not that it's not a reasonable question. It's not the problems that I can solve with my tool. Like, for instance, nutrition. There is no data that you can use. So eventually, through this kind of trying many things and failing all the time, eventually, we found an area with this radiology, and I should say Adam Yala, who is now a faculty and building his own company. Without him, I could not have done it because he started with me as an undergraduate researcher. I didn't have money to pay him and we didn't have data. I think I was really crazy or something because, changing your research career, even when you're a tenured professor, it's really not an easy task. Determination and belief really help. We really work together to make it happen. And slowly we moved into this task of building Mirai, and then we expanded to other areas.
Mm. And I wonder, so you've been working on Mirai. I think the first Mirai paper came out in 2019. You worked on it for a long time before that.
That's correct. The paper was ready like 2 years. We were hunting for data. So we were just hunting and begging. I really excelled in begging for data, for money. It was like my 2 years, like for all the sins that I've done. This was my atoning because I was constantly begging and asking and being nice. Then afterwards, when we did it, the doing the Mirai was actually relatively short. Most of the time then went constant rejection from all the journals. So we learned how to submit the papers. And I'm endlessly grateful to radiologists that accepted Mirai. But in 2019, there was the first publication.
Why was there so much skepticism at first?
So in terms, I think that the journals which are traditionally publishing clinical research, they have the certain mindset what constitutes a good paper. Like there are lots of researchers today, New England Journal of Medicine, main New England Journal of Medicine, recently published a paper that kind of says, we shouldn't be using AI tools because we didn't do prospective clinical trials. How naive the idea is, because if you're going to do prospective clinical trial on survival for 5 years using Mirai or whatever tool you are using, in 5 years, this Mirai will be obsolete, in contrast to drug, which is you cannot do retrospective study. For a lot of AI things, you need to do retrospective study. So many of these people are living in certain mindset, which is not really applicable to AI. And the same people who kind of judging traditional clinical paper now start judging other papers. So they didn't know how to read it. But I don't want to put all the blame on them. It took us a while to understand how do you communicate in the language and what do you need to show to be convincing. I cannot say that I perfected this art. I am better at it than I was, a few years ago, but still the bridge and the expectation, we're still working on it.
Okay. And something that's, I think, very striking about how you went about doing it was building this whole network of hospitals. And could you just say a few words about why it was so important to build that network?
So we were, first of all, I would say we're super grateful for Wellcome Trust and Bilal, who worked with us to give us, the first serious big funding in addition to Jamil Clinic, to really realize this vision that we don't want to develop AI tools and publish a paper and finish it here, but really bring it to the patient. Because the main reason for me for doing everything I do is to bring it to the patient. Because what I've seen, it left my head. I still remember it, and when I go to my checkups, I see these people. I really want to change their lives. And questions that you already asked me, how the doctors do it, because they cannot validate Mirai or Siebel on the spot. It's not like if you ask them, do you see cancer? And AI says yes, and they can verify. They can really see what happens in several years from now. So you want to make sure that it's for truly generalizable, because even in a place like MGH, there are many different types of patients from many countries because they serve, the Boston area. So what we've done, we created this organization where the hospitals around the world can come and join the network. And what it means that they can, Mirai is publicly available, so anybody can download it, but we can help them to bring it into their system, do whatever adaptations are needed. And we don't ask for their data, but what we do ask that they retrospectively validate it on their data and share these numbers with us in their own publication, doesn't have to be with us. And this is a first step towards translating it into clinical practice, because what we notice that depending how the screening, let's say breast cancer screening, works in your country, how you would use Mirai is different. Because in some countries, like in the US, you screen every year. In UK, you screen every 3 years. So the age when you start screening is different. So the AI tool would depend. How do you implement and adjust it? Where do you want to bring it? So our goal was first to see that it works across different population, and with Mirai and Siebel, we can say it does. And the second thing is from local clinicians to learn what is the best way to implement it in their healthcare system.
Just to close off this discussion, I'd like to end on a quick lightning round of questions in which you can just answer like in a sentence or two. The first question is, what's one problem that you're not working on that you secretly wish someone would solve?
This is not a very romantic and interesting and groundbreaking problem, but I really wish somebody to work on it, is figure out the economics of bringing AI to healthcare and to create the type of analysis and new tools which currently do not exist to combine together AI and economics. I'm just not educated this way.
Interesting, the whole cost effectiveness side of things. Yeah. Second question is, what's one thing that you would ban from medicine today, if you could?
I would not say to ban one thing. I would say an attitude that I would ban. And the attitude would be they see increasingly common across different healthcare systems. We've done it before. That's why we are right, which closes the opportunity to bring new technology. Among many other fields, we know that healthcare is really behind in bringing AI. And of course, there are many reasons, but I think this one, we've done it before, it works, is really the key reason behind this development, and I would ban it if I could.
And final question to end on a positive note. So, what's one thing that makes you very optimistic about AI in healthcare in the next decade?
So, what makes me optimistic is that finally I've started seeing that things are getting translated, because even a very determined person like me needs to see that this effort, besides producing papers, actually translated. So, one thing that made me super optimistic, Mirai, was a reason behind the change in NCCN guidelines, or some many main clinical guidelines on how to diagnose and treat cancer across all the areas of cancer. Based on the Mirai results, there was a change which allows to identify high-risk patients using image-based models. So it sounds to somebody like a tiny thing, but this is not, because it actually brings to the main healthcare realm a modern tool, and shows that you can eventually take your papers and take a hard research and bring it to change in clinical practice. So it gave me hope, and it also continued to fuel my determination to see this change not only in in Mirai, but in many other areas, and really to see that every patient would be having truly personalized risk assessment and treatment.
I think that's a great place to end. Thank you so much, Regina. This has been a pleasure.
Thank you, Mael.
Thank you all for listening to this episode, and I hope you enjoyed it. If you did, please subscribe and also share with a friend. I'd also like to say a special thank you to Gabriele Corso and Aziz Ayad, who helped me prep for this episode. See you next time.